Hello. So today, I want to ramble on a bit on optogenetics. It's basically a very new field in neuroscience that aims to incorporate light technology as well as genetics to further our understanding as well as improve research modalities of the central nervous system. So just to break down the terms, optogenetics, opto refers to light, optic, and genetics, well, genes. So researchers try to incorporate these two elements to produce a very, very promising modality. So a little bit of background. Our neurons, just like any other cells in the body, have a lipid bilayer, and embedded within these lipid bilayer are different types of channel proteins. There are voltage gated ion channels or ligand gated ion channels, so which basically means that these ion channels can be configured in a way to allow ions to enter when when certain things are bound to it. So for example, a ligand gated ion channel requires a ligand to bind to it first before its shape can change and thus allow ion entry into the cell. In the algae, a specific algae called Chlamydomonas Reinhardtii or something, I'll write it down later. So these algae, I think along with other photosynthesizing organisms as well. So these algae have light gated ion channels. Light gated meaning these channels can change its shape to allow ion entry when stimulated or when struck by light. So scientists, realizing the potential of these mechanism in the algae, have decided to incorporate such a thing in the human neurons. So what they did, they actually extracted the genes from the algae that encodes for the light gated ion channel and infused it into a virus. And then they used the virus to infect certain groups of neurons within the rat brain as well as the brain of a fly. So remember I said certain groups, right? So what they did in the rat, for example, was that they only infected the neurons in the central nervous system of the mouse that affects motor activity, affects their movement. So meaning a certain group of neurons in the rat's brain can now be stimulated by light, while the rest of the neurons that did not get infected by these genes, by the virus, only get stimulated by neurotransmitters. These group of neurons actually start producing action potentials and can actually start firing when a certain light frequency when they're exposed to a certain light frequency. So when the researchers shone blue light onto the rat, the neurons started firing. The neurons, that is, that encode for the motor function in the rat started firing. And what did you expect? The rat started running around. So the researchers then realized that they can actually control neuronal firing from afar with just light. So another thing that's super cool about optogenetics is that it allows only specific type of neurons to be involved. So like I said earlier, researchers can choose which group of neurons they want to infect with the gene, and only those neurons will be able to react to light. The others do not, like will not be able to react to light. And this is so important because the current modalities that we have in treating Parkinsonism, treating epilepsy, for example, we use something called the deep brain stimulation, which is basically electrodes implanted into your brains, and the firing of the neurons can be controlled as such from with electrical stimulation from the outside. The problem with this is that it's not specific to a certain group of neurons. So the neurons that are around the region also get affected, will also start firing unnecessarily. So therefore the patient will also eventually suffer from side effects. But with this optogenetic technology, only the neurons that we want to target and only the neurons that are involved in the disease will be involved. I mean, will receive the gene and will be stimulated. No other neurons would be affected at all. So the patient's side effects would be drastically reduced. So, besides just clinical potential, optogenetics is also being used to relate the cerebral function to human behavior. Specifically, a few researchers in the US are actually attempting to locate the seat of morality in the human brain. Seat of morality, people, I'm not kidding you. So it's a well-known fact that the limbic system is highly influential on human emotions. So knowing this, scientists are now trying to focus optogenetic technology onto studying that particular region in the human mind, to see human brain, to see if it does have a bearing on morality. And not just will the research be historical, because it's one of the first few researchers that used optogenetics, but it will also be phenomenal in terms of philosophy, because after all this time, we still do not know how we have morality, what is the need for morality, and even if all these questions have been answered definitively, we do not know where is it in the brain that uh, would would suggest a need for morality. So I'm actually a bit confused as to how they shall go about this. It's very interesting. It's also very, very promising, and I really hope optogenetics goes far. So that's all. Bye bye.